how would you distinguish monocot and dicot? So how can we differentiate monocot plants and dicot plants? So both monocots and dicots, they are flowering plants. They belong to the angiosperm division. Both plants produces flowers. But still there are differences, and the differences is in the morphology, that is root pattern, venation, flowers, cotyledons. These are the external features. And anatomy, if you take the internal structure, they differ in the number of vascular bundles in the stem, presence and absence of cambium, and the leaves. So let's learn the differences. So here, roots. In case of monocot plants, the root is fibrous roots, and in case of dicot, it is tap roots. So if you see the image here, it's the monocot plant and this is the dicot plant. Now in a monocot plant, it has got fibrous roots. Here the primary root which is formed is short-lived, and at the base of the stem many branches of root will be formed and they are arranged in the form of a network. So such kind of roots is called fibrous roots and it is found in monocot plants. But if we consider a dicot plant, so this is the primary root. It is long-lived and from the primary root many secondary roots will be formed. So this is called taproot system, and this taproot system is found only in the dicots. So this is a dicot plant and this is a monocot plant. So the first difference between the roots, monocot has got fibrous roots and dicots have got taproots. Coming to the next difference, venation. So in monocot plants, the leaf has got parallel venation. In dicot plants, the leaf have got reticulate venation. So monocot plants have got parallel venation and dicot plants have got reticulate venation. So let's see what is this parallel and reticulate venation. So here in monocot, this is the leaf and on the leaf the veins are arranged. Okay, the arrangement of veins are parallel to one another. They are straight. So this kind of arrangement is called parallel venation. And in case of dicot plants, leaf has got venation which is in the form of a network. So there will be many fine branches. So this kind of netted veins is called reticulate venation. So all dicot plants will have got reticulate venation and all monocot leaves will have parallel venation. So venation means the arrangement of veins, how they are present. Veins will carry the vascular bundle xylem and phloem to the leaves. So we learned fibrous roots in monocot, taproots in dicot, parallel venation in monocots, reticulate venation in dicot. The next difference is in the flowers. So flowers have got petals, calyx, corolla, androecium, gynoecium. Now these whorls will be present in the form of multiples of 3s in case of monocot plant. So we call it as trimerous. In case of dicot, it will be pentamerous or it will be multiples of 4. So the number of sepals and petals will be multiples of 3 in monocots and multiples of 5 in case of dicot. It is called pentamerous. In some cases, it will be multiples of 4. So here we can say in a monocot plant, there are 3 petals and always the sepals will be similar to that of a petal in the morphology. So 3 sets of sepals and 3 sets of petals. So they are multiples of 3. So totally here we can say 6. But in case of dicot, they will be multiples of 5 or in some cases it will be multiple of 4. So in this plant we can see there are 5 petals. So there are multiples of 5. So this is about petals. Even the sepals will be 5. And the next difference is between the cotyledons. So in a seed, so how many cotyledons will be present? And based on this structure, the division has been formed. So if it has got one cotyledon, we call it as monocot. If there are two cotyledons in the seed, it has got dicot. They're called dicot. Here is the seed. So here we can see a single cotyledon. So we call it as monocotyledon. So here a seed has got two cotyledon. So we call it as dicotyledon. So all dicot plants have got two cotyledons in the seed, and all monocot plants have got single cotyledon in the seed. Now coming to the anatomy, that is arrangement of the vascular bundles. If you consider the internal structure, when we take a transverse section of a stem, so we can see how the vascular bundles are arranged. In monocot, the vascular bundles are numerous and they are scattered. In case of dicot, they are present in the form of a ring. So let's see here. So here, if you consider the vascular bundle of a stem, there are numerous vascular bundles and they are just scattered in the stem. There is no proper arrangement. In case of a stem, dicot stem, the vascular bundles are arranged in the form of a ring. So here each one is a vascular bundle. They are arranged in the form of a ring. They are distributed in the form of a ring. So in monocot stem, it will be scattered. In dicot stem, it will be arranged in the form of ring. Now the next difference is cambium. So here in monocot stem, the cambium is absent. So that is why there is no secondary growth. In case of dicot, the cambium will be present. So that is why we can see a big trunk in the dicot trees. So cambiums are generally present in the vascular bundles. So it will be absent in case of monocot stem. In dicot stem, in between the xylem and phloem, there will be the presence of cambium. So in the vascular bundle, between xylem and phloem, there will be presence of cambium. So the next difference is the difference in the leaves. So in monocots, we can see the leaves which is isobilateral, and in case of dicot, it is dorsiventral. So here it is isobilateral. So the two sides will be similar. Here it is dorsiventral. So the upper and the lower portion will be similar. Dorsal and ventral will be similar in case of dicot. In case of monocot, the left and the right will be similar. So it is isobilateral. In case of dicots, it is dorsally ventrally similar. So these are the differences between the monocots and dicots. So in monocots, roots are fibrous roots. In dicot, it is taproot. Venation is parallel venation. Here it is reticulate venation. Flower, they have got trimerous, multiples of 3. Here it is pentamerous, multiples of 5. In monocots, single cotyledon. In dicots, two cotyledon. If you consider the anatomy, the transverse section shows numerous scattered vascular bundles. In case of dicot, 2 to 6 vascular bundles arranged in the ring. Cambium is absent in monocot. Cambium is present in dicot. Leaves are isobilateral in monocot, and in dicot it is dorsiventral. So these are the total difference present in monocot and dicot plants.